Hello, everyone. Today, I'm going to present my study of transcriptome and metabolic profiling of developing lentil seed. Lentil is an important legume species and a nutritional crop, providing a good resource of protein, minerals, and an array of diverse secondary metabolites, conferring the health promotion benefits. Seed development is a highly correlated process among different counterparts. These counterparts include the embryo, endosperm, and seed coat. It determines the structure and composition of mature seed and thus its end-use quantity. To understand the molecule and cellular events and the metabolic process during seed development is essential for improving lentil yield and seed nutrition value. In this research, we performed the transcriptome and the metabolic profiling for different compartments of development seed. First, we did the transcriptome analysis of embryo and seed coat for developing lentil seed. And to determine the corresponding stage, development stage, first we did the morphology study for the using a developing embryo for classifying the development stage of the seed. These the development stage include unfertilized ovule and two-cell zygote. Two-cell zygote is 48 hours after fertilization. Quadrant stage is 3 days, 3 days to 3.5 days after fertilization. Global stage is 7 days. Heart stage is 9 days. Torpedo is 11 days. Cotyledon early is 14 days. Cotyledon late is 18 days, and mature is 26 days. We used the lentiline CDC good in this study. Thus, that tissue study for transcriptome study used in is including unfertilized ovule, developing whole seed, and dissected development embryo, as well as the corresponding seed coat samples. After we did the transcriptome study, then we performed the profiles of secondary metabolites for the development seed, because that is the zygote stage and quadrant stage is the tissue limits. So we didn't for the secondary metabolites analysis. We didn't do these 2 stages. We did that for global and heart stage. We used the whole seed. In transition stage to mature stage, we did development embryo, development seed coat, and the whole seed. The secondary metabolites provide we did the following 5 metabolites analysis: the phytic acid, total phenolics, saponin, and soy aspirin, and isoflavones. After we have done the transcriptome study, first we did the principal component analysis and the sample to sample induced in sample distance study. From figure 1, we can see that embryo seed coat whole seeds, they are clustered in different clusters. So that's to say the transcriptome in development seeds different counterparts, showing quite different. Here is a heat map of embryo, essential, and seed coat related gene expression. From this heat map, we can say from A and B. A is embryo essential genes, and B is seed coat development related genes. And we can see that during the development stage, that is the embryo development and the seed coat, the transcriptome are quite different. After we did the transcriptome analysis, also we did the differentially expressed gene in embryos versus seed coat. For each stage, we for example, in zygote stage, and we compared that embryo and is corresponding to the same stage of seed coat. And for the all 8 stage, and the results showing that the differentially expressed genes are quite different between the embryo and the seed coat. We also did the expression analysis of genes involved in the flavonoid and isoflavonoid biosynthesis pathways and some other pathways for the secondary metabolites. In this figure, is showing that gene expression profiles in seed coat at different stage are illustrated with blue and red blocks, ordered from left to right. And we can see that in different stage, for flavonoid synthesis and isoflavonoid biosynthesis, these genes are activated or repressed differently. After we did the transcriptome study, then also we did an analysis of secondary metabolites content in embryo, seed coat, and whole seed. And in this figure, it's clearly showing that the metabolites in content in different embryo and seed coat stage are quite different. In summary, our study that in this study, we generated a high resolution of spatial temporal gene expression profiles in lentil embryo, seed coat, and whole seeds from fertilization through that maturation alongside the unfertilized ovule. We did a comparative transcriptomic analysis and with the gene co-expression network analysis, with embryo and seed coat, specify genes and the gene modules predominant in specific tissue and stages, highlighting the distinct genetic programs and between them. We investigated the dynamic profiles of flavonoid and isoflavonoid, phytic acid, serpin in different seed compartments across the K development stages. We identified a set of candidate genes involved in the biosynthesis of these metabolites. This study can providing the valuable resources for future studies to dissect the genetic contour of secondary metabolites to develop molecular tools for improving seed nutrition quality. If that's the study of the detailed study of this research. Thank you.